So nucleus, like we discussed earlier I think in the first class, was discovered by Robert Brown. Do you remember what did Robert Brown use for studying nucleus? Which plant was used? Robert Brown used which plant for studying nucleus? Anyone? Kedar, Siddharth? Robert Brown used which plant or which sample for studying nucleus?
Oak tree, I guess.
Oak tree was used by Robert Hooke.
Huh.
So that was for observing cells for the first time. So this is Robert Brown who discovered nucleus. So if you remember, he used orchid root cells, orchid epidermis. So orchid root epidermis is the outermost layer. So orchid root epidermis was used by Robert Brown for studying the structure of nucleus. And nucleus is the largest cell organelle in most of the cells. Inside the nucleus, you'll find something called chromatin material. So chromatin is the genetic material inside the nucleus. The term chromatin was coined by Fleming, Walter Fleming, not Alexander Fleming, Walter Fleming. Okay, so Walter Fleming coined the term chromatin, which is a material inside the nucleus that is a genetic material. We'll discuss later what chromatin is. Before that, let's discuss three important points: uninucleate, binucleate, and multinucleate. So Siddharth, what is the difference between uninucleate and binucleate?
Uni means one and bi means two.
Yeah, uni means one and bi means two. So most of the cells that we know, they are uninucleate cells. For example, amoeba is uninucleate, yeast is uninucleate. But there are some cells or some organisms in which they have more than one nucleus. So an organism with two nucleus is best example is paramecium. So paramecium usually has two nuclei. One is called the micronucleus, the other is called macronucleus. Micro means small, macro means large. Multinucleate condition is usually when the cell has more than two nuclei. For example, the cells of our bone marrow, striated muscles. So you've studied this in 9th standard, three types of muscles. Striated muscles are the skeletal muscles. So skeletal muscles are multinucleated, they have so many nuclei. Another one is most of the fungi. So several fungi and several algae also have more than one nucleus. So these 3 points are important. Examples of binucleate and multinucleate organisms. Next one, some of the cells of our body, they lack a nucleus. For example, erythrocytes. What are erythrocytes? Which cells are also known as erythrocytes? Or which cell of our body lacks nucleus?
It's a red blood cells.
Yeah, red blood cells. So erythrocytes are nothing but red blood cells. So red blood cells of most of the mammals, including human beings, and sieve tube cells of vascular plants. So sieve tubes are found in which part of the plant? Or which sieve tube is a component of which part of the plant? You have studied in lower classes about phloem. Yeah, it is phloem and xylem. So sieve tube is a part of phloem. It helps in conduction of food. So both sieve tube cells and RBCs of human beings, they lack nucleus, and such cells are called enucleated cells. So enucleated cells are those cells without a nucleus. So the reason why these two cells don't have a nucleus, at first they have a nucleus, but when they mature, they lack the nucleus. And you know that they have a very short lifespan, RBCs, so they don't need nucleus anymore. Their function is to just transport oxygen and other gases like carbon dioxide. So if they have to transport oxygen, they need more space. So that is why they don't have nucleus. Similarly, in sieve tube cells also, they have to transport a lot of food, and that is why they need more space. So as a result, they don't have most of the cell organelles. And nucleus being one of the largest cell organelle, if nucleus is removed, it will get a lot of space. Okay, so you should remember the first paragraph is very important. The name of the two scientists, Robert Brown observed nucleus in around 1831 in orchid root epidermis. The term chromatin was coined by Walter Fleming. So now I'll discuss the structure of nucleus in detail. What you see right now on the screen is how a nucleus looks like under the microscope. So you can see here, there is a spherical structure, somewhat spherical structure in the nucleus. The spherical structure is referred to as Nucleolus. So nucleus usually has one nucleolus, but even more than one nucleolus. Usually 1 to 4 nucleolus per cell. Then you can see nucleus is surrounded by a membrane. This membrane is called nuclear membrane, also known as nuclear envelope. This is also double membrane bound cell organelle. So nuclear membrane also is divided into outer membrane and inner membrane. Now the matrix of nucleus is called nucleoplasm. So you can say it is similar to the cytoplasm of a cell. Nucleus has a matrix, a jelly-shaped or fluid structure. So that is called nucleoplasm. Then there is chromatin material inside this. So somewhere here you'll find a network of chromatin material. This is the genetic material, DNA, which contains the genetic information. And also there are nuclear pores. So nuclear pores are small gaps in the nuclear membrane, which allows entry and exit of molecules. So let's read what is given in your textbook. The interface nucleus has highly extended and elaborate nucleoprotein fibers called chromatin. Now let's discuss what is interface chromosome. So interface nucleus is the nucleus of a cell when the cell is not dividing. So just before cell division, the genetic material will be found in the form of chromatin. So chromatin will be thread-like entangled mass of DNA. This is called chromatin. So this is just before cell division. Now, if the cell is going to divide or about to divide or the cell is dividing, what do you think will happen to this chromatin? What do you think will happen to this chromatin when the cell is dividing or about to divide? These will be converted to some other structure. Which structure?
Chromosomes.
Yeah, both of you are right. Chromosomes. Okay. So chromatin will be just before cell division. So when the cell is not dividing, it will exist in the form of chromatin. But when the cell is about to divide or when the cell is dividing, all this chromatin will be packed into structures called chromosomes like this. So DNA will be packed to form chromosomes. So chromosomes are formed only when the cell is about to divide or dividing. Okay. So chromatin is nothing but it is a complex of nucleoprotein fibers. Nucleoprotein fibers means it contains both nucleic acid and proteins. That is the meaning of the word nucleoprotein. Combination of nucleic acid and protein. Nucleic acids, which are the two nucleic acids? There are two types of nucleic acids. Which are the two nucleic acids?
Some ribonucleic acid and deoxyribonucleic acid.
Yes, ribonucleic acid and deoxyribonucleic acid, or in short, DNA and RNA. So DNA and RNA. So chromatin is made up of nucleic acid plus protein. That is why we use the word nucleoprotein fibers. And the nuclear matrix, then it also contains one or more spherical bodies called nucleoli or nucleolus. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes, so that is why we call it as double membrane bound, because the nuclear envelope has two membranes with a space in between, and the space in between is called perinuclear space. So there is a space between the inner membrane and outer membrane, and the space is approximately 10 to 50 nanometer. This value is important. You should remember. They will ask you in the exam, how much is the distance or how much is the length of the space, perinuclear space? That is approximately 10 to 50 nanometers. So perinuclear space is space between the inner membrane and the outer membrane of nuclear envelope. So this envelope, it forms a barrier between the materials present inside the nucleus and the materials outside the nucleus, that is cytoplasm. The outer membrane usually remains in continuous with the endoplasmic reticulum and might bear ribosomes with. This we already discussed when we discussed about endoplasmic reticulum, that is RER, rough endoplasmic reticulum. So rough endoplasmic reticulum is usually found attached to the nuclear envelope. At a number of places, the nuclear envelope is interrupted by minute pores. These minute pores are called the nuclear pores, which are formed by the fusion of its two membranes. The function of these nuclear pores is to allow the movement of some molecules like RNA and protein molecules in both the direction. So that means from nucleus to the cytoplasm or from the cytoplasm to the nucleus. So you should remember the function of nuclear pores. It will allow the entry and exit of molecules from the nucleus. Which molecules? Mainly RNA molecules and protein molecules. So right now what you see here is a detailed structure of the nucleus. You can see the spherical body here. The spherical body is the nucleolus. You can see thread-like structures, so many thread-like structures. These thread-like structures are chromatin. Then you can see nucleoplasm, which is the fluid or the matrix portion. Then you can see some gaps here and there. This is one gap. This is called the nuclear pore. There is a nuclear envelope, which is composed of outer membrane and inner membrane. And you can see here there's a space between these two that is called perinuclear space. The other things which are labeled in this diagram, you don't have to study. You'll study this next year in 12th standard. So you have to study only these four terms: nucleoplasm, nucleolus, nuclear pore, chromatin, and nuclear envelope. So if you're asked to draw the structure of a nucleus and label the parts, you should label at least these 5 parts, otherwise you won't get full marks. In NEET exam, the diagram will be given to you. It will be labeled A, B, C, D something. You will be asked to either identify the parts or they'll ask you questions about the parts.
So only these 5 parts are asked, right?
These 5 parts will be asked in NEET exam. I mean, if you're saying like with respect to 11th syllabus, this much is enough. You will study in detail about these terms next year again. So the next year there is an entire chapter about all these things, about genetic material, DNA. You'll study about genetics and molecular biology.
Yes, sir.
So right now this much is enough for you. So we'll discuss about nucleolus and nucleoplasm. So nucleoplasm, like I mentioned earlier, it is a nuclear matrix. Matrix is a cytoplasm kind of thing inside the nucleus. So nucleoli, the topic nucleoli or nucleolus is very important. So nucleoli, these are spherical structures which are found inside the nucleus. A very important point you should remember here is this one, very, very, very important. It is not a membrane bound structure. So since nucleolus is found inside the nuclei, it is not a membrane bound structure. And another important point, this point was asked in NEET exam, previous exam. What is the function of nucleoli? The function of nuclei is to synthesize ribosomal RNA, that is rRNA. So R is small letter here. The R here stands for ribosomal RNA. RNA is of 3 types. Again, this you'll study next year, but I'm just mentioning it now. RNA, which is ribonucleic acid, it is of 3 types. One is called mRNA, that is messenger RNA, which contains the information. rRNA, that is ribosomal RNA, which helps in the formation of ribosomes. Next one is transfer RNA, which helps in synthesis of proteins. So one of these called the rRNA, which is required for the formation of ribosomes. So ribosomes, when we discuss about ribosomes in the last class, I told you that ribosomes are made up of RNA and proteins. So that RNA is actually rRNA. So this rRNA is required for the synthesis of ribosomes. So we can say that if nucleolus is absent, will ribosomes be produced if there is no nucleolus? If a cell lacks nucleolus, what will happen to ribosome synthesis?
So synthesis of ribosomal RNA will stop.
Yes, synthesis of ribosomal RNA will stop. And if there is no ribosomal RNA, then there won't be any ribosomes also. And if there are no ribosomes, there won't be any protein synthesis inside the cell. So that is why nucleolus is very important. Nucleolus can help in the formation of ribosomes. So that is why this last point is given here. Larger and more numerous nucleoli are present in those cells which actively carry out protein synthesis. So if a cell is actively carrying out protein synthesis, that cell requires more ribosomes. That means the cell should have more nucleoli to form these ribosomes. So these 2 points which I've highlighted in green color, these 2 points are important. Nucleoli is not a membrane-bound structure. Second point, nucleoli is required for synthesizing rRNA or ribosomal RNA, which can be used to synthesize ribosomes. Then, next one. One second. Okay. So why do you think you look very similar to your parents, but you still have some differences? You don't look an exact copy of your parents, but you look very similar and you have some differences. And why do you think an elephant will always give birth to an elephant baby, not a lion's baby or tiger's baby, tiger cub? Why is it like that?
So because of transfer of genetic material.
Okay, because of transfer of genetic material. So you have heard about the terms heredity and variation,
Inheritance.
inheritance. So through reproduction, what happens is the mother's egg will fuse with the father's sperm, a zygote will be formed. The zygote will develop into embryo. So the zygote has chromosomes from both mother and father. One set will be from father, the other set will be from mother. So you have information from both the mother and father. So that is why you have some features of your mother, you have some features of your father, and you have some features which are not found in both your parents. So that is because of variations. The word variation means differences. Okay, so this process is called inheritance or heredity, by which genetic material will be passed on from parents to offspring. And the most common genetic material is DNA. So DNA will be passed on to daughter cells, not in the form of chromatin. It is usually passed on in the form of chromosomes. So let's see what is given in your textbook. So this we already discussed earlier. What is interphase nucleus? Interphase nucleus is when the cell is not dividing. So when the cell is not dividing, it has nucleoprotein fibers, a network of nucleoprotein fibers that is called chromatin. Now what happens if the cell is dividing? So during different stages of cell division, this chromatin will be converted, or it will undergo condensation to form X-shaped structures called chromosomes. So chromatin will become chromosomes during cell division. This is called DNA packaging. Now, if chromosomes have to be formed from chromatin, you have to take this thread-like chromatin and wind it around some round structures or ball-like structures, proteins. So those proteins are called histone proteins. Some basic proteins called histone proteins. What do you mean by basic proteins? What is the charge of basic protein? Is it negative or positive? When you say basic proteins, what is the charge?
So it is negative.
Negative. Madhura, what do you think?
Negative, sir.
Negative. Siddharth, are you there? Okay. So basic proteins are positively charged, not negatively charged. Acidic will be negatively charged. Okay. So acidic will be negatively charged. Basic will be positively charged. See, I'm assuming you guys have studied about COOH group in 10th standard,
Yes, sir.
carboxylic acid group.
Yes,
And
yes.
if I remove the H from this, it will be COO minus. So that is why acids will be negatively charged. But in
Yes.
case of positively charged basic proteins, they usually have the NH2 and NH3 group. NH3 group will have positive charge. The nitrogen atom will have positive charge. So you'll understand this better when you study in detail about this in chemistry. Also, we'll study about these charges in another chapter called biomolecules. So right now, just remember that these are basic proteins, positively charged. Now the question is, what is the charge of DNA? Charge of DNA, is it negative? Is it positive? Is it neutral? What is the charge of DNA?
I think it will be negative.
Negative. Kedar, what do you think?
So I think positive.
Why? Why do you think it is positive?
So because DNA has proteins, and here upper one you said basic proteins, they have positive charge. So I feel positive.
No, the proteins could be both acidic or basic.
Uh-huh.
So they are
Some,
acidic, right? So I guess they are negative.
which one is acidic?
So DNA.
Yeah. So you're right. We say, what is the full form of DNA? Deoxyribonucleic
Nucleic
acid, right?
acid.
Yeah. And I told you earlier, acids are negative. So of course, DNA should be negative. So this is negatively charged. Also, the main reason why DNA is negatively charged is because of the phosphate groups. So when we discuss about the structure of DNA later, I'll be mentioning that DNA is made up of phosphate groups. Phosphate groups you have studied in smaller classes, PO4 3 minus. So the charge of phosphate group is negative. So DNA is made up of lots of phosphate groups. Because of so many phosphate groups, the overall net charge of DNA will be negative. And that is why this negatively charged DNA can bind to positively charged proteins. You have studied earlier, negative and positive, they'll have a strong attraction. They are of the same charge, they will repel. So since they have opposite charges, they can bind or they can be bound to each other very strongly. And finally, they will form structures called chromosomes through condensation. So when chromatin, um, that is the DNA, along with some basic positively charged proteins, they bind together along with some non-histone proteins and RNA, it will undergo condensation and finally you will get something called chromosomes. So chromosomes are condensed genetic material. How many chromosomes do we have, human beings?
46.
Yeah, 46, that is 23 pairs. So why is it 23 pairs?
So
One
twice.
from
Yeah,
one from father and one from mother. So the pair
yeah, that is
contain.
right. So 23 you'll receive from father, 23 you'll get from mother. So that is why you have 23 pairs, total 46. Okay. Now, what is the approximate length of DNA? Suppose you take out a cell and you're going to stretch out all the DNA from one cell. How much will be the length of DNA from just one cell? Approximate length. In centimeters, how much will it be? So if you take out DNA from one cell, if you start stretching it out, it will be approximately 2.2 meters. That is how much centimeters?
220 centimeters.
Right, 220 centimeters. So it is given here in your book. A single human cell has approximately 2 meters long. So to be more precise, it is 2.2 meters. That is approximately 220 to 230 meters. Some textbooks say 2.3, some say 2.2. So in the next year, your NCRT will say 2.2 instead of 2. So we'll stick to that. So you have to stick to NCRT no matter what. There are lots of wrong information in NCRT. They won't change it, but still you have to study that only and you have to write that only because at the end of the day, so this has happened earlier also. Like there was a question in 2018 exam. Students were asked to choose which type of which form of iron, that is it ferric or ferrous, is absorbed by plant. So the correct answer is both. But since NCRT says I think ferric, you were supposed to write that. So students who wrote the correct answer, technically it is both. They did not get marks, but those who wrote what is given in NCRT, they were given marks. So CBSE will come up with an excuse that you have to refer this because it is given in NCRT. So for NEET exam, it is very important for both NEET chemistry also. So chemistry also, it is the same case. In NEET exam, almost all the questions from your chemistry book, chemistry paper will be directly from your book. You should study each and every line. They'll just change one of these lines into a question. So you should know each and every line in chemistry. So chemistry and biology. So in bio, you have around 90 questions. Approximately 80 to 85 questions will be exactly the same from NCERT. They will ask only like 3 questions out of NCERT. So you should read each and every line in NCERT. Then, so you should start reading it by now, because by the end, like you'll be writing your NEET exam in 2 years. So by that time, you will have 38 chapters in total, 38 chapters in bio, then approximately 30 to 32 from physics. So there'll be a lot of chapters, like more than 100 basically. So last topic is the structure of a chromosome. So this is the structure of a chromosome. You can see here, it is almost X-shaped. So this is when it is in condensed form during the cell division stage. Now you can see here, there is something in the center, a button-shaped structure in the center. The small constriction in the center is called centromere. So the function of the centromere, this primary constriction, its function is to just attach the two parts of the chromosome. So the chromosome has two parts. This is called the chromatids. Chromatid, not chromatin. This is called chromatid. So one chromosome, you can see here, it has two chromatids. So suppose if I cut this chromosome into two, you get two chromatids, one on the left side, one on the right side. Now, what are the other parts? You can see here, I've written something here, P and Q. So this was asked in 2019 exam, but this is not given in NCERT book. So let's see what is P and Q. P is the short arm of chromosome. So some chromosomes, you can see here, some of them have a short arm and a long arm. The short arm is called P arm and the long arm is called Q arm. The word P refers to petite, P-E-T-I-T. The word petite is a French word which means small. So that is why it is called P arm. And Q arm, just because the letter Q comes after P, so that is why they called it Q arm. Okay, so P arm is the short arm, Q arm is the long arm. Centromere is the central primary constriction which connects these two chromatids. Also, at the ends or the tips of these chromosomes, you will find a small cap-like structure which is not shown here. So this kind of provides stability to the chromosome structure. This is called a telomere. So telomere is like a cap and it protects the ends of the chromosome from getting cut. So telomere provides stability to chromosome ends. So these are the important parts of a chromosome. So chromosome has a centromere, it has a short arm and a long arm, it has two chromatids and it has a telomere. This is long arm. Also, one more point. On the sides of each of these centromere, you will find disc-shaped structures. These disc-shaped structures are called kinetochores. So suppose if this is a centromere, on both the sides of the centromere, you'll find disc-shaped structures. These disc-shaped structures are called kinetochore. Okay, so the function of kinetochore I had mentioned earlier. During cell division, spindle fibers will attach to the chromosome so that it can be separated. So spindle fibers will come and attach to these kinetochores. So kinetochores are the sites for attachment of spindle fiber during cell division. Again, we'll discuss in detail about kinetochore in cell cycle chapter. In this chapter, it is not very important. Now, you can see here 4 types of chromosomes are given here. So that is based on the position of centromere. What do you think is the first one? What is the position of centromere in the first diagram? This one. What is the position of centromere in the first diagram?
So I think it is fully grown.
Fully grown. No, centromere is the one in the center. So you can see here, position is exactly in the middle. So since it is exactly in the middle, this type of central chromosome is called metacentric. So metacentric chromosome is a chromosome in which the centromere is in the exact center. So both the arms are equal. There is no short arm or long arm. Both the arms are equal. So such a chromosome is called metacentric, centromere present in the middle of chromosome. In the next one, you can see here, it is somewhat slightly above from the center, slightly away from the middle of the chromosome. This one. This is called submetacentric. It's called submetacentric. In the third one, it is further away from the center. It is close to the tip, somewhat close to the tip, but away from the center. There is a short arm and long arm. This is called acrocentric. And in the last one, it is in the tip or the terminal region. And this is called telocentric. The word telo means tip or end. Telocentric. So based on the position of centromeres, you can classify chromosomes into 4. First one is called metacentric. So that is centromere in the center, middle. Submetacentric, centromere is slightly away from the middle. Acrocentric, centromere is far from the middle and it is close to the tip. And telocentric, it is at the tip or the terminus. Okay, so you should remember these shapes. You should be able to draw these shapes in your board exam. And if you're given these diagrams, you should be able to identify which one is which. Also, you can see here on the first chromosome, metacentric chromosome, there are 2 round structures at the top. So these 2 round structures at the top is called a satellite. Okay, so this is called a satellite. This one is called a satellite. So it is a secondary construction. So satellites are secondary constructions which are non-staining. So even if you stain a cell or a chromosome during under the microscope, satellites, you won't be able to see satellites clearly. They're non-staining secondary constructions. So you should remember these 2 words. What is a satellite? Non-staining secondary constructions found at the tip of chromosomes.